Welcome to How to Choose a Spatial Biology Platform. Spatial biology is the study of cells in their native tissue context, and it was originally invented in 2019. Since then, it's been widely adopted by many fields such as oncology and neurology. The first question to ask yourself is, what is the goal for your research? Is this a discovery project where you don't really know what you're looking for, you're just hoping to find things? In that case, you definitely want to cast the widest net possible. And that means doing something like Visium HD or the new HD3 prime so that you can capture the whole transcriptome. In contrast, if you have an idea of what changes your treatment is going to induce, which pathways, for example, then you may want to go with the off-the-shelf, more mid-plex panel. And you can even add into those. What level of resolution do you need? Does it have to be true single cell, or will subcellular whole transcriptome be sufficient? Again, what species are you working with? Although all Xenium, Cosmix, and Visium have panels available for human and mouse, they're not widely available for other species. That said, the new Visium HD3 prime is a poly-A capture method, so it'll work on all eukaryotes. Another potential limiting factor is how were your tissues prepared? Xenium, Cosmix, and HD are all validated for FFPE and fixed or fresh frozen tissue. The new HD3 prime, however, is only validated on fresh frozen tissue at the moment. And I'm sure 10x Genomics is working on many other kinds of tissue types. And then when we come to data analysis tools, how comfortable are you doing data analysis? Xenium offers Xenium Explorer as a free download from its website, and it's an open platform compatible with common tools such as Seurat and SquidPeep. Visium HD and HD3 prime have Space Ranger, whose output can then be uploaded into Loop browser. The image on the right is actually from Loop browser. And then again, what's your budget like? How many samples are you going to test? How large are your samples? All of the platforms have different scan areas. Xenium has the largest. One thing you may consider is that you may be able to put more than one sample within the scan area as long as they don't overlap or cover the fiducials. And that concludes our introduction to choosing a spatial biology platform.